Alright, this is Jahangir from MedPorium, and today we have part 3 of our lymphatic and immune system unit, in which we will discuss adaptive immunity. Now, adaptive immunity, or specific immunity, is a second type of immunity in the immune system with two characteristics. One, it is specific, meaning it recognizes and directs an action against a particular pathogen, and two, it has the ability to memorize a pathogen previously fought. This ability of memorization helps the body react to the pathogen at a much faster pace. Adaptive immunity is split into humoral immunity or antibody-mediated immunity and cellular immunity or cell-mediated immunity. We will discuss each later, but to put in layman's terms, humoral immunity employs antibodies that are either secreted by B lymphocytes or circulating the bloodstream, and these antibodies attach to the antigen of a foreign pathogen to tag them as the enemy for T lymphocytes. Cellular immunity employs lymphocytes, primarily are T lymphocytes that directly attack and destroy a pathogen. Before we move on, we need to discuss the two different forms of immunity, and that includes active and passive immunity. Active immunity is when our own body brings us immunity through the creation of antibodies and lymphocytes, and passive immunity is when we receive immunity from the antibodies and lymphocytes of someone else. Both forms can be further differentiated to natural or artificial immunity. Natural active immunity involves a production of antibodies and lymphocytes from our own body to bring us immunity against a pathogen that has been exposed to the body. Artificial active immunity involves vaccination as a means to provide aid for our immune system. Natural passive immunity involves a fetus receiving immunity from the mother through the placenta. And finally, artificial passive immunity, which is the injection of antibodies created from another person or animal. Now let's discuss antigens. Antigens are molecules that can trigger immune response and help the body identify intruders. Within these antigens are structures known as epitopes. Epitopes, also known as antigenic determinants, are a proportion of the antigen an antibody will bind to. Antigens are characterized by self-antigens and non-self-antigens. Self-antigens are antigens presented in the cell within our bodies, and these antigens are antigenic to other individuals rather than ourselves. Antigens are conveyed on the major histocompatibility complex, or MHC proteins, which are found in many of the cells of the body. There are two groups of MHC proteins. The first is known as MHC1, which are found in nearly every cell in the body and convey a self-antigen of the body. The second is known as the MHC2 proteins, which are found in the antigen-presenting cells as well as other cells involved in the immune response. MHC2 conveys the antigen of a foreign pathogen. Non-self-antigens, as the name implies, are the antigens of foreign pathogens that the immune system will attack. These antigens can also be complete antigens and incomplete antigens. Complete antigens differ from incomplete antigens in that they can issue an immune response by itself. Incomplete antigens such as haptens are too small to be immunogenic by itself, but when they bind to a large carrier such as a protein, they can issue an immune response. Now let's discuss the primary lymphocytes of adaptive immunity, and they fall into three categories: T lymphocytes, B lymphocytes, and antigen-presenting cells. The T lymphocytes, as we discussed, originate from the red bone marrow and migrate to the thymus for maturation. They are born from hemopoietic stem cells. Precursor T cells enter the thymus through the corticomedullary junction, as this portion of the thymus contains numerous blood vessels. The T cells will then migrate to the subscapular area of the thymus and develop T cell receptors, or TCR for short, CD4 receptors, and CD8 receptors, which are negative at first. Once this process occurs, thymocytes will test the T cells in a process known as positive selection, which occurs in the thymus cortex. In positive selection, the thymocytes will introduce a foreign pathogen to the self-MHC protein. If the T cell cannot recognize the self-MHC protein, the cell will experience apoptosis or cellular death. And if the T cell can identify the self-MHC, this results in MHC restriction, in which the cell is restricted in recognizing the antigen of a cell MHC protein and moves on to negative selection. Negative selection occurs in the thymus medulla, in which T cells must not recognize the self-antigen of the body cells. CD4 and CD8 receptors become positive. CD4 positive cells are introduced to MHC2 proteins, while CD8 positive T cells are introduced to the MHC1 proteins. If the CD receptors recognize the self-antigen, they experience apoptosis, which is crucial in eliminating T cells that may cause an autoimmune disorder. Once the T cells have passed positive selection and negative selection, they leave the thymus through the corticomedullary junction and into lymph nodes and other secondary lymphatic organs as naive T cells. which are just T cells that have just matured and that have not encountered an antigen before. Now, before we discuss B lymphocytes and their development, we need to discuss antibodies. Antibodies, also known as immunoglobulins, are defensive gamma globulins that bind to the antigen and neutralize a threat. Let's look at the anatomy of an antibody. The antibody has 4 polypeptides, 2 which are known as the heavy chain and the other 2 are known as the light chain. Both chains are held together by a disulfide bond. Each chain will have a variable region which combines to form the antigen binding site of the antibody. Each chain will also have a constant region which determines the antibody's class or mechanism of the antibody. There are 5 classes of antibodies: immunoglobulin alpha, immunoglobulin delta, immunoglobulin epsilon, immunoglobulin gamma, and finally immunoglobulin mu. Now let's discuss the development of B cells, and that begins in the red bone marrow. In the red bone marrow, stromal cells will release signals towards the lymphoid progenitor cells, which are just our stem cells, starting the B cell development process. Right now, the heavy chain genes and the light chain genes are both germline, meaning they have an unaffected, unarranged genetic material. Here's what that may look like. We have a variable, diversity, and joining genes of the heavy chain of the antibody, followed by the variable and joining genes for the light chain segment of the antibody. Remember, the heavy chain of the antibody will contain the genes V, D, and J, while the light chain will contain the V and J gene. Both chains will have a constant gene. In the early pro-B cell stage, D and J join together, and in the late pro-B cell stage, V joins with DJ. Now we move on to the large pre-B cell stage, in which our VDJ complex will rearrange to form the heavy chain and bind to the surrogate light chain. This whole complex is known as our pre-B cell receptor. After proliferation of the large pre-B cell, we enter the small pre-B cell stage, in which the variable and joining gene of the light chain join together. Our variable and joining gene will then rearrange in the immature B cell stage, in which our immunoglobulin Mu will be expressed on the cell surface. In mature B cells, immunoglobulin delta will also be expressed on the cell surface. These mature naive B cells will leave the red bone marrow and enter the secondary lymphatic tissues, where they will be ready to attack a pathogen. The last cell we will discuss is known as the antigen presenting cell. Now, throughout this series, we have discussed the antigen presenting cell from a definition standpoint. Let's discuss how the antigen presenting cells function in providing antigen. That starts by phagocytosis of a foreign antigen, in which the antigen will be degraded in the cell. The antigen residue will then be released through exocytosis, with some fragments of the antigen that has been degraded by enzymes being displayed on the cell surface through the MHC proteins. These fragments are known as epitopes, and this is the part of the antigen our T cells will bind to and recognize. Alright, and unfortunately that wraps up part 3 of our lymphatic and immune system unit, in which we had an introduction to adaptive immunity. In this introduction, we discussed the functions of adaptive immunity, the different forms of immunity, antigens and antibodies, and the primary lymphocytes of adaptive immunity. And as always, a Pomodoro day will make you doctor someday. I'll see you next time.